Hi, everyone. So let's get started for session 4, Stem Cells for Precision Platforms. Today, our moderators are going to be Dr. Noor Mustafoglu and Dr. John Clegg, both from the Wyss Institute. So if you guys want to share your screen.
All right, great. Thank you, Naveed, for the introduction, and we're happy to moderate this next session on stem cells for precision platforms. And we have a great lineup of 4 speakers here today. Again, my name is John Clegg from the Wyss Institute at Harvard University, and I'll be introducing the first 2 speakers. And so our first speaker for today is Dr. Olia Bela. She is from the Gladstone Institute and will be presenting a talk titled Engineering Human Neuromuscular Circuits. And so with that, I'll turn over the table to you for your talk.
Hello, thank you, John, for the introduction. So today, I'm going to be talking to you guys about neuromuscular circuits. So neuromuscular circuits are essential for voluntary movement and involve multiple neuronal and non-neuronal cell types. And the basic neuromuscular unit is the neuromuscular junction, which is formed between a motor neuron and a skeletal muscle fiber. The neuromuscular diseases affect, such as ALS and SMA, affect one or more of these cell types, and this results in motor deficits. So because of the complexity of these circuits, it's very hard to determine the contribution of individual cell types to these diseases, especially when working with complex systems such as animal models. On the other hand, conventional, traditional in vitro systems normally are just co-cultures in which synapses are formed randomly, and this makes hard to extract quantitative data from these systems. So our goal was to engineer controllable neuromuscular circuits that provide functional readouts and use them to get a better understanding of these diseases, and also as drug screening platforms. So we decided to start with the most basic unit, which is the neuromuscular junction between muscles and motor neurons. We had 3 requisites for this system. We wanted it to mimic human physiology. That means we wanted our system to be 3-dimensional and compartmentalized. So what we envisioned was a skeletal muscle tissue growing between pillars, and then separately an aggregate of motor neurons that will be able to grow axons and innervate this tissue. The second thing, we wanted the system to be controllable. For that, we decided to make our neurons photosensitive. So to do so, we use the channel rhodopsin protein, which is an ion channel that opens in response to blue light. By introducing this in our motor neuron membrane, means we can stimulate action potentials just by shining blue light on them. And the last condition, what characteristic that we wanted from our system is to get functional outputs. So that's what's achieved, because if the neuromuscular junction is functioning correctly, this action potential from the neurons will be transmitted to the muscle, and the muscle will contract. And we can easily detect and quantify this using optical tools. So this photosensitive motor neuromuscular junction is basically the central piece of our approach. And then it gets slightly more complicated just because we want also our system to be human and patient or donor specific. And then we want it to be quantifiable. So to achieve the human specific, patient specific system, we start from a skeletal myoblasts, which are primary cells. And what we did is we reprogrammed the cells into iPSCs. Then we made these iPSCs optogenetic, introducing the channel rhodopsin. And then we differentiate them into optogenetic motor neurons. So both cell types have the same genetic background. And then we introduce these 2 cell types in our advanced cell culture platforms, and we establish these photosensitive neuromuscular junctions. So to make the system quantifiable, then what we did is just build an optical stimulation platform and combine it with custom video processing. And with this, we can score and quantify the function. So I'm gonna go into a little more detail about these 2 pieces. So this is the culture system that we use initially. This is a microfluidic system developed in the Rogers Camp lab at MIT. So it has two different like channels to see the separated muscle and neurospheres. So these channels are connected to these two chambers. So the one for the muscle is provided with pillars for attachment. And then there's a second chamber that can house a neurosphere and then allow the axons to grow and innervate the tissue. So it looks like this, the whole process. You see individual myoblasts in the muscle chamber, and then you induce differentiation or myotube fusion. And then after 2 weeks, you get a strip of muscle growing between both pillars. Separately, outside of the system in a suspension culture, you induce differentiation of the channel-relapsing iPSCs to get channel-relapsing motor neurons growing as a neurosphere. So at day 15, we see one of these neurospheres in each of the devices, and then allow the axons to grow and innervate of muscle. And this is how it looks after 2 weeks of culture. You can see this is like half of the tissue. This is where the pillar is. You can see how the axons innervate the tissue and also the striations of the muscle. So we had our neuromuscular junctions, and now we have to like build a system to be able to stimulate them precisely. So we built this optical stimulation platform, which I won't go into many details, but basically it's just two Arduinos controlled by two LEDs controlled by Arduino microprocessor. So one is blue, and this is the one that's going to be stimulating of neurons, and the other one is the red one that's just providing bright field illumination. The one thing I want to point is like here, like after the blue light gets to the sample, but before it gets to the camera, we have a filter that's blocking it. And so that means like we won't be seeing the flashes of light in our movies. This is just to make our video processing easier. So this is actually the output that we get. So as I mentioned, we don't see the flashes of light, but we do have this blinker that indicates where the LED is on. So the software basically generates these trays, which measure the contraction of the muscle. So each of these peaks is a muscle contraction. And then what it does is correlates these peaks with like the light pulses, which are the vertical lines. So if there's like a contraction right after a light pulse, it gets marked as an effective pulse and it's shown in green. And if there's no, then it gets marked as red and labeled as an effective pulse. So that what we basically use is like this fraction of the pulses that actually result in a contraction as a way to evaluate the functionality of our tissue. And the only other thing that we need to take into account is because there's some spontaneous activity of the muscle already, we just calculate the chances of a contraction just happening right after a light pulse. So this is just a small correction that we do to like get our final score. Using this system, we were able to like measure changes in the neuromuscular junction. And one of the first things we observe is that the function of our neuromuscular junctions improved like over time. So here we can see movies from the same tissue at day 9 after the start of the co-culture and a week after. So you can see how it goes for barely being responsive at all. Actually, the score was very, very low. And then after 7 more days, it gets much more responsive. So this is something that we observe in all our tissues. It's basically, there's 2 weeks of like improvement in the function and then it kinds of plateau. What is interesting here is like by following the same tissue over time, we are able to see and measure and quantify gradual changes, not just like classify them as like responsive or not responsive. So then we move on to do some disease modeling. So we were interested in myasthenia gravis, which is the most common disorder of neuromuscular transmission. And it happens because the patients develop antibodies that attack or block the neuromuscular junctions. So this was interesting to us because it means like theoretically we could get a serum from these patients that contains these antibodies and introduce it in our system and recapitulate this disease. So that's what we did. So we had our tissues, split them in 3 groups of like similar function before the treatment. So we are going to not treat the first group, treat the second group with myasthenia gravis serum, and the third group with healthy serum. So we treated the tissues for 48 hours, and we saw like the myasthenia gravis group just stopped responding completely. And this is one of those tissues. So as I said, like before the treatment, it was like very responsive to light, and then after the treatment, there's just no response at all. So what we did next is like we wash out this serum and let the tissues recover for another 48 hours. And we did a partial recovery of the function. So this is interesting, and it's telling for two reasons. Like the first one is the recovery just happened in only some of the tissues. So that like shows us that like the response to these antibodies, or the nature of the antibodies in the serum from patients, is like different. Some of them are binding reversible, but some of them are maybe destroying the neuromuscular junction, and that can be reversed. And this is something that has been observed in patients. The other thing is like this whole process kind of mimics what happens in patients when they go through a myasthenic crisis, and the symptoms get very, very bad, and they get put into plasmapheresis, which basically is just washing out all the antibodies from their blood, and then they improve. So I want to talk about other things that we are like doing now, and like the avenues that we are pursuing with this. So first, as I mentioned at the beginning, we are interested in building complex circuits, so not just the neuromuscular junction, but also other cell types. And we can do this in many ways, because as I mentioned, there's many cell types involved. But we are very interested on the interneurons, because interneurons are the ones giving the input to our motor neurons. And in a lot of diseases, it has been shown that like motor neurons are malfunctioning. And this could be because they're dying or the disease is affecting them, but it could also be due because they are already receiving the brown signal from their input from upstream. So our lab has a protocol to differentiate these excitatory interneurons from iPSCs. So we are now working on like modifying our system to basically introduce interneurons. So these interneurons are now going to be the ones that are photosensitive, and they are going to innervate our synapse with our motor neurons that we are introducing a calcium reporter on them. So we can see the functionality of the motor neurons with this calcium reporter, and then we can still see the contractility of our muscle. And we are very interested in looking at this in the framework of disease. So we can like basically swap which elements are diseased, which ones are healthy, and get a better idea of like the role of each of these cell types in the disease phenotype. The second thing is like we are part of the somatic cell genome editing consortium. So we are now using these systems to evaluate in situ gene editing. And so we are like trying to, we're trying to see like not only that we can correct whatever mutation we're trying to correct with gene editing, but also that this leads to like better function. And also, we are also interested in see what's the undesired effect of gene editing that could also be affecting function. And then the last thing is like we are continuing our studies with myasthenia gravis, and we are working with a pharma company actually to do some drug screenings. So this is it. I want to acknowledge my former lab, in which like most of this work was done. Also, obviously, for organizing this. My current lab, the McDavid lab at Glasgow Institutes, and finally, yeah, my former and current collaborators, as well as funding sources. I'll be happy to take any questions.
All right, great. Thank you so much for a very, very interesting talk and cool platform. We have a number of questions rolling in. And so I'll start with the first one. This question is from Ankit Kumar, who asks, do you observe any muscle fatigue when frequent stimulation of your model?
That's a very good question. Yes, we actually do. So that's why that's the reason why we separate normally measurements that long, like 48 hours or 24 hours at least, because if you try to like just do this repeat measurement several times, you will see fatigue. And that's also the reason why we choose to do this kind of ramp stimulation, where we increase the intensity of the frequency of our stimulation in one measurement, so we can like test at the same time, like how many times it can respond and like how fast it's able to respond.
Great. And then so another question that came in from Yvonne Lamelli. asks, have you tried combining different types of cells on the muscular model, endothelial, for instance?
No, I haven't. But so the lab that developed this platform, the MIT, they actually have done something with endothelial cells, and I think it improved the muscle functionality.
Great. And then one final question is from Moshe Wilner, who said, thank you very much for your talk. Can you please explain how you introduced the CHR2 to the motor neurons?
So we have done this in several ways. Like we have used lentiviruses, and we also have done like just CRISPR. So we have different lines with, and we do this in the iPSCs, and then we just differentiate them to motor neurons.
I see. Okay, and actually I have one more question, which asks, are you using any animal-derived products in the construction of the model?
No. No. Well, no. Not that I know about now. No, I don't think so.
All right. Well, great. Well, thank you so much again for your talk. And I guess in the interest of moving on and staying on time, we'll move to the next speaker. But thank you for your talk. And I'll go ahead now and introduce our next speaker. And so the next speaker in this session is Dr. Anna Sisolak from Columbia University, who will be today giving a talk which is titled Modeling of Fibrotic Lung Disease Using 3D Organoids Derived from Human Pluripotent Stem Cells. And so I welcome Anna to share your screen and go ahead and start your talk.
Okay. Okay, can you see well my screen?
Yes, we can see it.
Perfect. Okay, so let's start. First of all, I would like to thank you very much for a kind introduction and for this opportunity to present our recently published work focused on human lung organoids for modeling human fibrotic lung disease. So briefly to introduce the concept, the pathologies of the respiratory system are among leading causes of morbidity and mortality worldwide. They are actually characterized by limited number of effective medical therapies, a low number of lung transplants for qualifying conditions, and actually in many cases, a poorly understood pathogenesis. So development of more effective treatment is hindered by the lack of preclinical models of human lung that could fully recapitulate human lung development, capture on all the complexity and heterogeneous phenotypes of the disease of interest, together with the providing a source of cell-based targets for therapies or disease prevention. In this context, direct differentiation of human pluripotent stem cells, which are unspecialized cells that can give rise to specialized cell types, has a high potential to deliver such highly needed models. So going to the next slide, we can see that the protocol that our lab developed is for differentiation of human pluripotent stem cells, leading to specification of lung field, which allows to grow in suspension so-called lung bud organoids. Once those LBOs are embedded in the matrigel, they undergo branching morphogenesis and model human fetal lung development. To further apply our LBO system for respiratory disease modeling, we focus on idiopathic pulmonary fibrosis, IPF. So IPF is a progressive and ultimately fatal disease, which is characterized by the fibrotic obliteration of lung alveoli, leading to respiratory failure. The therapeutic options are extremely limited, with lung transplantation being the only definitive currently treatment for the patients. The etiology and pathogenesis of IPF are unclear, but age, environment, and genetics are the most commonly known risk factors currently. IPF is also believed to be caused by repetitive alveolar epithelial cell injury and dysregulated repair. Those last evidences came from subset of patients with Hermansky-Pudlak syndrome, HPS, which is similar clinically entity with IPF, and is caused by abnormal biogenesis and trafficking of lysosome-related organelles. Those patients are characterized by pigmentation and bleeding abnormalities associated with dysfunction of melanosomes and platelet granules, respectively. On the other hand, those are also similar to the of alveolar type 2 cells, where surfactant is stored and recycled. The mutation causing HPS affect 4 distinct protein complexes. Out of 10 known mutations, only HPS1, 4, and 2 are associated with features of pulmonary fibrosis, and those we called Hermansky-Pudlak syndrome interstitial pneumonia, HPS-IP. The fact that only some HPS mutations are associated with pulmonary fibrosis offer us a unique opportunity to determine features of epithelial cells that are shared by those with mutations that predispose to HPS-IP. So what we actually did, we introduced IPF-associated mutations and those that are not predisposing for IPF to embryonic stem cells using CRISPR-Cas9 gene editing technology, and characterize HPS mutant lung organoids morphologically and with transcriptomic and phenotypic approaches. So now the results of our studies, first focusing on the morphological changes. Here you can see the bright field images of day 40 matrigel embedded lung organoid culture that was generated from parental Y type and HPS mutant lines. Those cultures demonstrated that HPS1, 4, and 2, but not HPS8, shown the abnormal compacted morphology. We further characterize those organoids with mesenchymal and extracellular matrix markers, and we compared immunofluorescence staining in one Y-type and mutant organoids. Those data revealed increased deposition of collagen 1 and fibronectin and increased presence of cells expressing mesenchymal markers such as vimentin in HPS1 to 4, but no HPS8 lines. Here I'm just showing 3 representative markers, profile for 3 markers, just for the simplicity of the talk. But we screen more markers that are described in the paper. Those observations we just saw in the immunofluorescence staining, they were further confirmed with quantification and RT-qPCR. So in contrast to HPS 1, 2, and 4 mutant lines, no differences were observed in HPS8 mutant organoids compared to the Y-type, which is in line with the clinical profile of HPS-IP patients. Since the pathogenesis of IPF and HPS-IP involves epithelial injury, we perform RNA-seq on purified epithelial cells from organoids that are generated from Y-type and each particular mutated clones to identify gene expression signature that differs from parental line, but have some shared profile in the fibrotic organoids. So a total of 56 genes were found to be significantly upregulated in both HPS1 and 2, but not HPS8 organoids compared to the Y-type profile, while 28 genes were significantly downregulated. Screening the list of the genes that we obtained, among upregulated one, interleukin 11 was of particular interest for us, as it is a soluble cytokine. We actually decided to further characterize the effect of interleukin-11 in our system. We found that increased expression of interleukin-11 was observed in fibrotic-prone conditions, which we quantified by RT-qPCR. On the protein level, we perform ELISA in the supernatant of HPS1 organoid culture that was increased. And finally, we also quantify, we also determine the expression, the increase in expression of interleukin-11 in IPF and HPS-IP patient samples by in situ hybridization. And finally, just to determine whether interleukin-11 can drive fibrosis in our model, we culture the Y-type organoids in the presence of or absence of human interleukin-11. Organoid treated with interleukin-11 shared increased expression of mesenchyme markers by immunofluorescence, and here confirmed with RT-qPCR. As a final point, we deleted interleukin-11 in HPS4 knockout fibrotic prone lines, and those organoids displayed a morphology that is comparable to Y-type model. We confirmed that, those results in immunofluorescence and again in RT-qPCR. All those results suggest and confirm a central role of interleukin-11 in pulmonary fibrosis. So in conclusion, what we found and what we describe in the paper is that HPS-associated fibrosis can be modeled in embryonic stem cells derived lung organoids, and that interleukin-11 is a potential regulator of fibrosis in this context. And the second part of the results actually is in line with recently published data. Those data started appearing last year and keep coming over this year, that interleukin-11 is a potential target for pulmonary, cardiovascular, and liver fibrosis that is actually emphasize more the effect of those cytokine for potential targeting in fibrosis. Okay, so with that, I would like to thank you and acknowledge all the members of our lab, especially our advisor Hans Snook and Alex Strikodis, who performed most of the experimental part for this project. Also, I would like to thank the funding that we get to perform this work. Thank you very much for your attention, and I'm happy to take some questions.
All right, well, thank you very much for sharing what was a very interesting talk, and we have a number of questions pouring in. So the first one is from Sarah Cuevas Ocana, who asked, how did you quantify the expression of the ECM proteins to determine an increase in the fibrotic prone mutants? Did you perform flow cytometry, or did you estimate this from the immunofluorescence images?
We performed the quantification from the immunofluorescence staining. Also, we have for selected markers, we also performed the Western blot, but most of the profile came from the staining.
Okay, great. The second question is from Stephanie Campbell. She asks, does interleukin-11 also have a role in initiating fibrosis in other organs, the liver, for example?
Right. So what I mentioned by the end of my talk, this cytokine is starting to become more and more known in terms of fibrosis, and there are more work in different organs that provide the support for those data. So not only our group found the important role of interleukin 11 in the lung, but it also demonstrate in the cardiovascular and liver conditions. So, right, it's becoming more and more interesting target for fibrosis, right?
Great. The next question is from Liam Reed. Liam asks, were any markers of endoplasmic reticulum stress and unfolded protein response upregulated in the derived type 2 alveolar cells of the HPS mutant organoids?
I probably had some of those markers upregulated, but I don't have a list right now, and I would say probably if they were upregulated, the level of expression was not that much pronounced, that we didn't really focus on the first step to analyze those markers.
Okay, and so one final question that I had was, in your RNA-seq analysis where you identified up and down regulated genes, did you notice any, I guess, mechanistic type insights from the ones that you did not investigate further, so the ones other than IL-11?
So we focus mostly on IL-11, but yeah, we could group the upregulated, downregulated genes in several categories. So there were some that actually belong to the group of extracellular markers regulation. Those that were, let me check if I can find, I probably have a slide. A slide over here. Yes, so I have a small table here. Actually, I don't remember by memory, but yes, if you can follow this slide, we have the group of genes that were related to the extracellular space, the secretory molecules, plasma membranes, cell surface genes. So there's a couple of groups that we can define, but we didn't investigate much further those.
Okay, well, great. Well, thank you very much for what was an excellent talk and very insightful response to many questions. Looks like we do have one more very specific technical question in the Q&A that I'm going to ask you to address via comment in the Q&A box.
No problem.
And with that, I'll hand over the session to Nur to moderate the last two talks. Thank you.
Thank you.
Hello, everybody. My name is Neuromuscular. I'm also from the WIS Institute, working with the Dr. Ingber's group on the brain targeting program. And it's actually next speaker will be also talking about blood-brain barrier, which I'm really interested in. And it's my pleasure to introduce July Linwell from Johns Hopkins University. And his talk will be titled as Tissue Engineered Models of Blood-Brain Barrier Tight Junctions During Health and Disease.
Thank you for the introduction. Can you hear me?
We can hear you.
Great. Okay, so thank you, Nur, for the introduction. Today I'll be speaking about tissue engineered models of the blood-brain barrier tight junctions during health and disease. One of the time-lapse images that I'll talk about more is shown on the right here. So the adult human brain is comprised of over 100 billion neurons, and these neurons are supplied by nutrients from a 600-kilometer network of microvessels. And if you actually zoom in on these microvessels, you see a cross-section that's shown here, where we have brain microvascular endothelial cells that are then surrounded by other cells, including pericytes and astrocytic end feet. And together, these three cell types form what's known as the blood-brain barrier. And a really important function of this blood-brain barrier is to then regulate what can enter into the brain parenchyma. And this acts to protect neurons from dangerous compounds, but also then is a direct barrier to restricting or a direct barrier to allowing drugs to enter into the brain. And one of the main reasons why the barrier is formed is through the expression of tight junction proteins. And these proteins act as a physical zipper between adjacent brain microvascular endothelial cells, blocking the paracellular pathway of transport between the endothelium. And compared to peripheral endothelial cells, the brain endothelium is particularly enriched for these protein components. And so in general, we're very interested in looking at tight junction proteins because disruption of these proteins is associated with aging, neurodegenerative disease, brain cancer, stroke, trauma, and many other brain diseases. So to study tight junctions, there are some existing models, including knock-in mouse models. However, these obviously lack human specificity. And then there's also other techniques, including electron microscopy and immunocytochemistry, but these have poor temporal resolution. So for this work, the goal is to build three-dimensional human blood-brain barrier models that have very high spatiotemporal resolution of tight junctions. And to do this, we're utilizing induced pluripotent stem cells, which we can differentiate into brain microvascular endothelial cells. We then also apply these cells within models that are able to mimic physiological cues like cylindrical geometry, shear stress, and cell ECM interactions to actually build 3D systems that resemble the in vivo blood-brain barrier. And for the work today, I'll be specifically focusing on brain microvascular endothelial cells, not as much the other supporting cell types. So once we actually create these 3D models, we can then use real-time imaging to study tight junction behaviors. And today I'll talk about some applications of this model, including looking at how tight junctions were modeled during the cell cycle, how tight junctions repair following physical injury, and mechanisms of oxidative damage. And all these questions are very relevant to studying how tight junctions change in the blood-brain barrier, as this is an important process that happens during many brain diseases. So to form our models, we're utilizing iPSCs that have green fluorescent protein tag zonula occludens 1. ZO1 is a scaffolding protein that connects the transmembrane tight junction to the actin cytoskeleton. And using these cells, we derive brain microvascular endothelial cells using existing differentiation protocols. And at the end of this process, we achieve blood-brain barrier phenotype characterized by expression of some blood-brain barrier proteins, including tight junction proteins like ZO1, Claudin 5, and occludin, endothelial proteins like CD31, and then nutrient and efflux transporters. Additionally, if we look at the gene level, we see that differentiation is associated with the loss of thousands of genes, some of which include pluripotency markers, and a gain of thousands of genes, which are associated with blood-brain barrier phenotype. Additionally, we can then actually confirm functional behaviors of these cells by recording transendothelial electrical resistance. And we see that cells have very high electrical resistance compared to many other cell sources. So using these cells, we combine it with some microfluidic devices where we can suspend a 150 micron diameter rod within a PDMS microfluidic device. We then around this rod gel collagen hydrogel. When the rod is then removed, it leaves a bare channel that can then be processed with crosslinkers and ECM components before we then add our cells, which then assemble into a three-dimensional tubular structure. We can then perfuse this tubular structure at constant shear stress over a week's time to study aspects of the model. And then specifically with these cells, we can conduct epifluorescence imaging to look at tight junctions in real time, or confocal imaging to look even closer at real-time tight junction dynamics. So this three-dimensional model of the blood-brain barrier has been characterized for phenotype in many ways. One of the ways is through looking at permeability of dyes. So we see if we have a non-brain specific microvessel formed with HUVEX, you have a very leaky endothelium to this dye lucifer yellow, while in our brain microvessels, it completely restricts transport of this dye across the endothelium. Additionally, the model displays localized tight junctions through immunocytochemistry, and also unique gene expression, where we have around 100 upregulated and 100 downregulated genes compared to our 2D models. And if we look at the genes that are upregulated, These are associated with processes including extracellular matrix organization, angiogenesis, leukocyte migration, among many others, kind of suggesting that our three-dimensional model will have unique responsiveness to many perturbations compared to 2D systems. Additionally, we can combine other cells into our models, including iPSC-derived pericytes, which become located on the abluminal side of the brain microvascular endothelial cells. So using this model with the Z01 tag cells, we can conduct several types of experiments. One simple thing we can do is just look at tight junctions in real time. So we can characterize cells in microvessels as being apoptotic, mitotic, or quiescent. So from dozens of hours of imaging of microvessels, we can look at around 2,000 cells, and we see there's this really small fraction of cells that are actually apoptotic and mitotic compared to most cells which are quiescent in the model. Interestingly, we've actually found that the rates of apoptosis and mitosis can be related back to the average cell area within the microvessel. So if you have a microvessel with very large cell area, mitosis dominates. Well, if you have a microvessel with really small cell area, apoptosis dominates. This suggests that there's some sort of homeostatic cell size in which turnover of the endothelium will be balanced between these two opposing processes. Shown here at the bottom then is some GIFs of these different processes over 1 hour of imaging in the microvessel. And we can trace different cell types and then also their neighboring cells to look at behavior of these cells. So from this imaging, we can identify morphological changes that occur across the cell cycle. We can extract tons of metrics on each individual cell. We see that quiescent cells have very stable metrics. So these properties are only oscillating by about 5% from baseline values during imaging. There's some things associated with apoptosis that are quite obvious, like a loss of cell area and perimeter, but also other changes like gain in circularity and instantaneous velocity that are associated with this process. If we look at the cells neighboring apoptosis, we see actually that they're quite stable in area. This means that the loss of a cell from the microvessel is not directly compensated by its cell neighbors, but instead compensated by the entire endothelium, not just its direct neighbors. But interestingly, there's actually decreases in area after apoptosis. For mitotic cells, we can see the formation of two daughters. We see that area and perimeter increase of these cells after division, while cells neighboring a mitotic cell are very stable and resemble that of quiescent cells. And so these parameters are important to characterize before we look at some perturbations to the model. So the blood-brain barrier can be physically damaged in several cases, including things like trauma, stroke, and traumatic brain injury. So to kind of model this physical injury, we've developed a laser ablation technique to selectively ablate around 10 endothelial cells from the endothelium. When we do this, there's then an immediate increase in permeability at that site, which we can visualize with this dye Evans blue. And then we can image in real time the recovery from this ablation. We see over 2 hours the closing of this wound. And so the main result I want to talk about here is that this recovery process is completely dominated by cell migration, not by proliferation of cells. So the closing of this wound is from cells migrating inward, not any sort of division occurring at these boundaries, which contrasts some results from two-dimensional wound healing assays. Additionally, we've used this model to look at how tight junctions respond to oxidative stress, which is a cue that's associated with many brain injuries and disease. So we can dose this oxidative agent metadione and in real time look at tight junction dynamics as well as barrier function. We see that perfusion of this oxidative agent causes endothelium detachment. So we hypothesize that this is related to breakdown of focal adhesions or basement membrane. And while this process occurs, we still see that there's many intact tight junctions. So this compound isn't necessarily directly affecting the tight junctions as much as the macroscopic adhesion of the microvessel as a whole. So to summarize, we've developed three-dimensional human blood-brain barrier models with high spatiotemporal resolution over tight junctions that enable studies of the BBB during health and disease. If we compare some of these results to more standard in vitro models, we see some unique advantages of using a 3D system, as well as some unique findings compared to kind of the standard in vitro systems. And with that, I'd like to thank the Searson group as a whole, and then specifically some excellent undergraduate students, Matt, Alana, and Gabby, who have heavily contributed to this work. With that, I can take any questions that people have.
It was a great talk. Actually, we have several questions. Let's start with the questions from Roger Cam. He says, great presentation. Can you tell if the vessels become leaky at the point of an apoptotic cells?
Thank you for the question. So we do not see any leakiness at sites of apoptosis.
And another question from Alp Özgün. Can you mention how you're able to the IB max to model injury, especially in such a controlled manner?
So I think I didn't completely understand the question, and I can't see it on my screen. It's how do we?
Yeah, can you mention how you're able the IB max to model injury, especially in such a controlled manner?
In terms of the laser ablation, perhaps? Yeah, so basically we can direct the laser ablation to the bottom plane of the microvessel and have certain conditions to reliably ablate a specific area of the microvessel. Hopefully that answers the question.
And another question from Detlef Obel. Did you try different surrounding cells or any genetic modification of the iPSCs?
Yep, so we have models with pericytes, isogenic pericytes even, that have just a different fluorescent tag to look at how pericytes influence the function. In terms of genetic differences, we also have work looking at how different neurodegenerative mutations affect the properties of the brain endothelium.
I actually have a related questions. Did you try pericytes model when you applied the oxidative stress? Because it looks very interesting. Pericytes may eliminate the attachment of the IPS BMX.
Yeah, we've not done that, but I think that's an excellent point that the pericytes could be kind of strengthening the basement membrane or supporting in other ways.
And we have one more question from Victor Oja. The repair to injury due to the cell migration, could this an effect from perfusion?
Very much so, that's possible. So because traditional wound healing assays, I mean, some can incorporate shear stress into them, but it's possible in our system that because of the size of it and the different shear stresses that contributes to the lack of, to it being dominated by cell migration.
And we have two more questions actually, but I will ask you to answer those later with a comment if it is possible, because we need to move on with the next speaker. And we will continue with Andrew Daly from the University of Pennsylvania. And his talk will be titled 3D Bioprinting of High Cell Density Heterogeneous Tissue Models Through Spheroid Fusion within Self-Healing Hydrogels.
Hey everybody, are you able to see my screen?
Yes, we are.
So that's okay. You can see this slide correctly?
Yeah.
Okay. First of all, thank you, Pamela and NIBIB, for organizing. It's been a great symposium so far. I hope I can keep people interested. This is the last talk before the panel. So I'm going to talk about our recent work on bioprinting of high cell density heterogeneous tissue models through spheroid fusion in self-healing hydrogels. So as we all know, cells possess remarkable capacity to self-organize into multicellular spheroids, including organoids in vitro. And for example, starting from embryoid bodies, we can use these techniques to emulate organs such as, for example, brain organoids or cardiac spheroids. And the advantage of these approaches are that we can develop models that have organotypic cell densities. As a result of this, we have high levels of cell-to-cell and cell-to-ECM interactions. And these systems are capable of supporting developmental like sorting and differentiation, which makes them extremely promising for modeling aspects of human development and disease. However, obviously, they're very small and miniaturized models, and this can make it quite challenging to control cell and ECM patterning across larger length scales, which may be interesting in other aspects of and other questions related to human development and disease in vitro. So we're interested in bioprinting in the British Lab, and there has been some previous work done on developing bioprinting techniques specifically for spheroids and potentially organoid type structures. So for example, here you can see the Kenzan method, where cell aggregates are aspirated with a microneedle and then transferred onto these needle-like arrays for controlled placement and fusion into 3D constructs. More recently, a more gentle method has been developed where cell aggregates can be aspirated. Then using a hydrogel printer, hydrogel can be deposited in a 2D surface. And then into this hydrogel, we can control, it's possible to print spheroids within 3D space. And this process can be repeated in a layer by layer fashion. So these systems are really promising for developing constructs composed of multiple spheroids and controlling placement and local heterogeneity. However, there is quite a lot of mechanical disruption with existing methods that can impact cell viability and function. And in the 3D printing field, there has been an increased interest in developing gel-in-gel printing techniques. So rather than, say, traditional printing where a gel is extruded onto a two-dimensional surface, in these cases, the gel is actually printed into another material that we call, in this case, a self-healing material. So here you can see extrusion of an ink labeled red here into this secondary material in blue that locally fluidizes and self-heals around the printed nozzle and extruded filament as we print. And this provides extra support, allowing us to build up more complex structures. In the Burdick Lab, we're very interested in hyaluronic acid, and specifically, we previously developed a guest host chemistry to create self-healing hydrogels. So the hyaluronic acid is modified with adamantane and cyclodextrin, guest and host molecules. They have an affinity for each other, and when come into contact, they will form a gel. However, this crosslink is relatively weak, and under shear, this crosslink will break open. However, it is reversible, so it will reform. So we can see it possesses these nice self-healing properties, such as our shear thinning behavior and self-healing. And so what does it do? So here you can see a nice video of printing of a biomaterial ink into our support hydrogel to create this complex spiral-shaped structure using an extrusion printing technique. So we started to ask the question whether we could use this kind of platform to develop a method for printing of cell spheroids within 3D space. So we adopted this technique where, like before, you can see in this video, we were able to aspirate a cell aggregate in a media reservoir. Then, due to the self-healing properties of the gel, we were able to transfer it into our into the gel. You can see it shear thinning around the needle. And then we were able to deposit the cell aggregate within 3D space to build up arrays of spheroids within 3D. And then using this technique, just some examples with mesenchymal stem cell spheroids. We can print these rings, layers of rings to create pyramid and cone shaped structures. We can also print spheroids of distinct composition to, for example, potentially control the local cell type or the local ECM type within a tissue. So having established that we could precisely position these cell aggregates within 3D, we wanted to determine whether they could actually fuse after we print them over culture. And we were able to show that when we deposit this ring of aggregates over 4 days of culture, they fuse together into a continuous ring. So importantly, the cells are not interacting with the gel here, so there's no adhesive ligands, and it's viscoelastic, so they're able to displace and push the gel away, and instead they're predominantly fusing with each other. And we can build these structures up into, for example, tube-like constructs composed purely of spheroids. So having established that we could print these microtissue models, we started thinking about developing a platform for disease modeling and drug screening. In particular, we've been interested in cardiac tissue in the Burdick lab, and cardiac spheroids have been used quite a lot in the field as a platform for drug screening. So for example, using pluripotent stem cell derived cardiomyocytes or cardiac fibroblasts, and in some cases endothelial cells. These cells can be mixed together in different ratios to create miniaturized models of cardiac tissue. Because of the high levels of cell-to-cell contact within these types of tissues, it's a promising cardiac microenvironment, and we can see nice staining for sarcomeres within this tissue. And in addition, it's also been shown that the addition of these supporting cardiac fibroblasts can improve cardiomyocyte maturation. So, and because of this, these systems are very promising as a platform for screening different drugs for efficacy. So we began to ask the question whether we could create a model of focal cardiac scarring. So post-MI or infarction, due to focal and local cardiomyocyte death, their fibrotic scars form within the cardiac tissue. So how can we use our printing technique to create a model of this interface to look at interactions between healthy and scarred tissue? So to do that, we formed healthy and scarred cardiac spheroids by mixing different ratios of iPSC-derived cardiomyocytes and fibroblasts. So a 4:1 ratio for healthy microtissues and a 1:4 ratio for scarred microtissues. As expected, the healthy spheroids contracted a lot more than the scarred ones and displayed higher levels of calcium activation. And then using these two distinct populations of spheroids, we could use these as an ink to create rings of cardiac tissue. So 8 healthy spheroids to create a purely healthy ring, or 7 and 1 scarred spheroid to create a scarred ring. And you can see nice confocal imaging of this focal region of high fibroblast density. And when we looked at the functionality of these tissues, physiology using calcium imaging, first looking at these sequential frames of a single calcium activation event, we can see that in the healthy samples, we can see this with this change in fluorescence from blue to a more yellow-like shade. We can see a relatively uniform activation across the tissue, indicating a healthy phenotype of cardiac tissue. However, when we incorporated the scars, we can see in the central region, we get this delayed activation with initial activation in this bottom side, and then a delayed activation occurring over after the scar. And we were able to quantify this by mapping our activation delays to confirm that the incorporation of these scars significantly disrupted activation within the tissues. So having established this scarred microtissue model to mimic cardiac tissue, we started thinking about how would this be useful for a drug screening question. And in our lab, we've been previously interested in techniques that can induce cardiomyocyte proliferation. Inherently, probably as most people know, they have a relatively low capacity for proliferation, especially in the adult. And microRNAs that target the Hippo pathway have been shown to induce cardiomyocyte proliferation, potentially offering a solution for generating fibrotic cardiac tissue. Previously in our lab, we've delivered this microRNA in a rodent model in rats, and just here looking at the functional outcomes after a month, we can see that this microRNA is capable of restoring the ejection fraction compared to controls. And however, there have been more recent scaled up studies, for example, in pigs, looking at a very similar microRNA that also targets the Hippo pathway. And although the ejection fraction can be restored, it can also induce significant complications at later time points. So this indicates that the survival of the pigs beyond that 1 and 2 month period, showing that actually almost 70 to 80% of the pigs died due to uncontrolled cardiac failure. So it's clear that these Hippo inhibiting microRNAs are promising. However, dosage and timing clearly must be very tightly controlled before further translation. So we were interested in screening this microRNA using our microtissues. So initially, just looking at scarred spheroids, we looked at how the treatment duration, so either 2, 4, or 7 days delivery could influence cardiac output. We looked at the contraction amplitude at 2, 4, and 7 days over time and found that a 4-day period was necessary to induce a significant increase in the contraction amplitude, with no further increases at 7 days. And then when we looked at microRNA delivery for 4 continuous days on these printed rings, so using that same system we had before of printing a cardiac microtissue with a scar that's got a significant activation delay here, we could show that when we treated these microtissues with microRNA, this activation delay could be attenuated or improved, resulting in increased synchronization within the tissue. Again, we can also look at this using histological staining for cardiomyocytes in red with troponin and fibroblasts with fomentin. And using our EDU label for cell proliferation, we could see co-localization indicating cardiomyocyte proliferation within the scarred regions. So with that, we can conclude that our self-healing HA hydrogels can support precise printing of spheroids in 3D space and then fusion into 3D microtissues. This approach is generalizable and can be implemented with a wide variety of spheroid and organoid models. And we believe this opens up a lot of new opportunities in printing of precision models to mimic diseases and for drug screening. So with that, I'd like to thank the Burdick Lab. I'm happy to take any questions now, but here's some contact information if you need it. And we recently uploaded a preprint on this work, so you can find more work there, or information there if you're interested. So with that, happy to take any questions, and thank you for listening.
Thank you very much, Andrew. This was very interesting and very informative talk with great pictures and videos. And actually, it was interesting for our audience too. You got so many questions. We only have time for a few of them. So let's start with Mona Mansouri. Have you ever tried to make spheroids with more number of cells? Why only 4,000 cells per spheroids?
So we have created 10,000 cells for spheroid. We kept with 5,000 to I guess limit the effect of diffusion limitations. So not to have them too large that we will be getting a necrotic core within larger spheroids. And I guess we were able to answer our question, we think, with the smaller spheroid size. But it would be possible to go larger depending on the question that was being answered, or if you're interested in creating a larger piece of tissue for implantation.
One more question from Pancarbo. How many cycles of cells healing can you undergo in the hydrogel without changing its structural properties?
So previously, we have demonstrated that they can support a significant amount of shear thinning and reversing of that shear thinning. So I can't give an exact number, but I mean, in a single larger printing technique, we can do this up to maybe 30 or 40 times, and we don't see any problems there. So yeah.
And maybe one last question from James Armstrong. Is this first printing process automated or is it manual?
So we're using a robotic system, but there is, it's not fully automated yet in terms of positioning. So we're using a joystick, but currently we are working on automation under, so James, I don't know if you're familiar with the Osbalock group, they are working on that too. So it is definitely possible to fully automate, but yeah, we're working on that.
Thank you very much. This was very interesting. There are more questions are rolling in. I will kindly ask you to, if you can answer those questions in the Q&A, and that would be great.
Yeah, thank you.
And it is my pleasure to moderate this section with John Clegg. And all the talks was very interesting and informative for all of us. And I would like to thank to all you guys.
Great, thank you to our moderators and our speakers. This was a great session on stem cells. We have time for a quick 3-minute break before we begin our 5th and final session with talks today, and session 5 will be on cancer models. So we'll just take a few minutes to get our speakers set up.